All right. So what we'll do next is just mention some kind of miscellaneous things about enzymes that will help in understanding how enzymes work. One miscellaneous thing about enzymes is this. So say that this is an enzyme. Its active site, again, is shaped so that it can only accommodate a certain shaped reactant. It turns out, though, that after a reactant, after a substrate binds to its enzyme, the enzyme ends up clenching even tighter after that interaction happens. So after the substrate binds to its active site, the enzyme kind of snuggles up to it, clenches onto it even tighter. And so we'll jot this down here. So after a substrate contacts the active site, that enzyme clenches even tighter to that substrate. And this is called an induced fit. Right? So after the substrate binds to its active site, it induces an even tighter fit around that substrate. The reason why this induced fit happens is this. So again, enzymes are proteins, and that means that there's all kinds of R groups lining this active site of the enzyme. Some of those R groups are polar. They have a partial charge to them. And so if this enzyme ends up contacting a substrate that also has partial charges on it, you might remember that when polar molecules get close to one another, a type of bond forms, a hydrogen bond. And so it's these hydrogen bonds that end up kind of pulling this active site in closer around where that substrate is. Right, so the idea here is that induced fit, they occur due to hydrogen bonding between the substrate and the active site's R groups. And again, that hydrogen bonding is what causes that more snug fit to occur. Here's another thing to know about enzymes. It turns out that oftentimes enzymes need a little help in order to work properly. So in this picture, this represents an enzyme, this represents its substrate, and what we mean by help is this. It turns out that sometimes other molecules have to kind of occupy the active site in order for the enzyme to bind tightly to its substrate. So these helper molecules in red, there's two general classes of enzyme helper molecules. There's some that are called cofactors, and what cofactors are non-protein molecules that help enzymes bind to their substrate. Another general class of helpers are called coenzymes. Right? So if it's an enzyme helper that happens to contain carbon, then it's called a coenzyme. So this is a little bit confusing the way it's written here, but cofactors and coenzymes, they're the same thing in that they both help substrate bind to its enzyme. What makes the cofactor different from the coenzyme basically is what it's made of. Cofactors are inorganic. In other words, cofactors are molecules that have no carbons in them. Coenzymes are organic molecules. In other words, coenzymes are molecules that have carbon in them. So this kind of illustrates one of the reasons why things like vitamins are really essential in the diet. So vitamins don't really give you energy. Instead, the reason why they're important is because vitamins are actually used in order to make cofactors and coenzymes. So if you stop eating vitamins, you would run out of cofactors and coenzymes, which would mean that all the enzymes in your cells would eventually stop working. And if all the enzymes stopped working, that means that the chemical reactions that are happening inside of your cells, the ones that are keeping you alive, those would slow down to a really annoyingly slow pace, a pace that would not sustain life. Right? So vitamins are important. One last thing we'll mention about enzymes is this. So it turns out that the enzymes in your cells, they're not on all the time. So sometimes it's a good idea to turn off an enzyme. So for example, if you have a chemical reaction that's occurring inside of a cell and it's already produced enough product, you'd probably want that chemical reaction to stop, right? Because it's kind of a waste to keep making something that you already have a lot of. So again, it's often a good idea to shut down enzymes every now and then. And the thing that our cells use, the things that our cells use to shut down enzymes are inhibitors. And so generally what an inhibitor is a molecule that prevents an enzyme from binding to its substrate. And what we'll show you here is that there's two general types of inhibitors. They work in two different ways. One type of inhibitor works like this. So say that this is an enzyme. Here's its active site. And here's the substrate for this enzyme. One class of inhibitor is called a competitive inhibitor. And this competitive inhibitor is represented by this red triangle here. And the way that it prevents a chemical reaction from occurring is that you can probably see where this is going. This inhibitor here has a very similar shape to the enzyme's normal substrate, which means that this inhibitor could just as easily fit into the active site of that substrate. When that occurs, it would prevent the real deal from fitting in the active site. And so because the substrate can't bind to the enzyme, that substrate cannot be converted into product, so no chemical reaction occurs. This inhibitor basically is a dud, right? Its shape makes it so that it can fit into the enzyme, but it's different enough from the substrate so that it actually does not undergo a chemical reaction. So whenever you have an inhibitor that works like this, it's called a competitive inhibitor. Right? So it might make sense why it's called competitive, because it competes directly with the substrate for that spot, for that active site. Right? Whichever one of these two gets there first, that's going to be the winner. They're competing with one another. Right? So competitive inhibitor competes with the substrate for a chance to bind to the active site. It turns out that there's another general class of inhibitor. So there's another way that a molecule can prevent an enzyme from binding to its substrate, and here's how. So again, in this picture, this represents the enzyme. This represents its substrate. This represents another type of inhibitor called a non-competitive inhibitor. And so you might be wondering, well, how can something like this, something that looks like this, prevent this substrate from binding to its active site here? Here's how. The way that this type of inhibitor works is that it doesn't bind to the enzyme's active site. Instead, it binds to some other spot on the enzyme. And whenever that occurs, that causes a change in the shape of this enzyme so that the substrate can no longer bind to its active site. Right? So it's just kind of another way in which enzymes can be prevented from binding to its substrate. Because this type of inhibitor doesn't really compete directly with this substrate for the active site, right? They're not aiming for the same spot. They're called non-competitive inhibitors, and again, they work by binding to a different spot on the enzyme. When that occurs, that causes the active site of the enzyme to change shape. And when the shape of the protein, when the shape of the enzyme changes, the substrate can no longer bind to its active site, right? That specificity, that the matching shape is kind of destroyed after this point. One thing to mention here is this. So you see this spot right here, this spot on the enzyme where the non-competitive inhibitor binds to. It's called the regulatory site, or in other places you might see it being referred to as the allosteric site. Right, so again, this thing right here where the non-competitive inhibitor binds to, it's called the regulatory or the allosteric site.